Hello students, welcome to EPG Patashala. I am Dr. Gayatri, Assistant Professor in Center of Advanced Study in Crystallography and Biophysics, University of Madras. Today we are going to learn the module on crystallographic databases which comes in the paper Biocrystallography. Let's see what are the objectives we are going to learn in this module. We are going to cover several databases that are used to deposit the structures of small molecules as well as the macromolecules. The first and foremost is the Cambridge Structural Database, which is used to deposit small organic and metallo-organic compounds. Inorganic crystal structure database is used for deposition of powder diffraction files, and there are several other databases like the American Mineralogist Crystal Structure Database, Mineralogy Database, MINDAT, and Database of Zeolite Structures, ASM Alloy Phase Diagram Centers, and for macromolecules, that is for deposition of protein structures, RCSB PDB is very famous and most frequently used. Nucleic acid database is used for the deposition of DNA structures, and there is also a combined chemical dictionary which we will be seeing in the further lecture. The objectives of the module deals with the various databases as we saw earlier that are available for the deposition of small and macromolecular crystal structures. The structural information of the molecules are stored and maintained in these different databases which are regularly updated, and the curation of the data submitted is conducted regularly. The data submitted to these various databases are characterized and different statistical information of the data are provided. Now let's see what is a Cambridge Structural Database. Cambridge Structural Database, which is generally called as CSD, is a world's repository for the deposition of small molecule, organic and organometallic crystal structures. It is the most widely used resource in structural chemistry, which is distributed by Cambridge Crystallographic Data Center, which is termed as CCDC. This was established at the Department of Chemistry in Cambridge University, which collates and makes the CSD database available. The major objective of CCDC is the advancement and promotion of science of chemistry and crystallography for the benefit of the public. It has built up considerable expertise in various research aspects, including drug design, crystal engineering, molecular structure analysis, and also knowledge mining. It celebrated 50 years of sharing the crystal data in the year 2015. Olga Kennard began collecting the crystal structures in the year 1965 in believing that the collective use of the experimental data would lead to the discovery of new knowledge which transcends the results of individual experiments. CCDC plays a major role in the collection of about 8 lakhs of compounds and making them available to the scientists all over the world. Each and every structure that is deposited in the CSD consists of the chemical information, the experimental details, the physical properties of the compound, the structural details, and also the bibliographical information. WebCSD is the online portal of Cambridge Structural Database, which contains few lakhs of small molecule structures, as we saw earlier, more than 8 lakhs of structures. It provides easy and convenient access to the wealth of the structural knowledge. Website is another database which is also maintained by the CCDC. It is used to search the data that are deposited in the CSD by using any of the search parameters like the author name, title of the compound, in which year the data has been deposited, what is the name of the compound, etc. CCDC offers several products and services with few restricted to licensed institutions, and other modules and products are free of charge for the benefit of scientific community. These products and services cover a wide range of crystallographic tools, starting from the data collection, validation, visualization, teaching, research, and also for analysis. We can see an exponential increase on the number of structures that are being deposited on CSD every year. since 1972. As we saw, there are several CCDC products and services that are available. One of the important program is NCIFER, which is used to check, edit, and visualize the crystallographic information file that contains all the details about the structural information. MOGL is another program to validate the molecular geometry. It provides an easy access to information on the preferred values of bond length, angles, that is valence angles, acyclic torsion angles, by using the data derived from the CSD. Prequest is a data conversion program with an objective to create high quality structural data files in the format that is searchable by Conquest. Quest is another program which is used to search the entries, either 1, 2 or 3-dimensional searching of the CSD in the format that is searchable by Conquest. Quest is another program which is used to search the entries, either 1, 2 or 3-dimensional searching of the CSD. Conquest is also for 1, 2 and 3-dimensional searching of CSD, but with graphical interface. Conquest, in addition to structure drawing, it provides a full range of text and numeric search options. For example, one can search by using the author name, the journal name, the name of a compound. One can use the formula, molecular formula, phase group, unit cell, and so on. Mercury is a crystal structure visualization and exploration tool. It offers a new collection of tools for the analysis of geometrical, chemical, and crystallographic data. It also provides interactive statistical, charting, and plotting options alongside the 3D structure visualization and also analysis. To check the unit cells against the CSD during data collection, CellCheck CSD will be used. Web Deposit is a web service that enables the submission of ZIP files and related structure factor files for storage at CCDC and also for subsequent deposition in the CSD. Generally, CCDC uses two different codes for the deposited ZIP file. One code will be assigned for the validated and deposited CSD entry with the deposited number in a format CCDC and followed by 6 numeric values. The second code is for curated CSD entry, which will be assigned a 6-letter reference code. In order to obtain a crystallographic data from a CSD, one can use the search called as a request a structure. Dash is another tool. It is an indispensable interactive and accurate tool for solving the structures of simple to complex compounds from powder diffraction data. WebCSD is an online portal to Cambridge Structural Database to provide easy and convenient access to the structural data that is deposited in CSD. WebCSD is restricted to only licensed organizations and institutions. To do a statistical analysis and to do a graphical display, Vista has been used. CSD symmetry is a collation of observed molecular and crystallographic symmetry properties. One can also use the CSD teaching subset. It contains number of examples including BSc PR and stereochemistry modules. CSD Solid Form module provides knowledge-based informatics tool for improving the effectiveness, quality and risk assessment in the development of solid formulation of drugs, agrochemicals and molecular materials. RelayBase is a tool to provide easy access to search, retrieve and analyze protein ligand complexes. IsoStar is a CCDC's library of intermolecular interactions. It is a web application that provides thousands of interactive 3D scatter plots in order to show the probability of occurrence and spatial characteristics of interactions between the pairs of chemical functional groups. Each scatter plot shows the distribution of a contact group, say for example a OH donor group, around a specific central group, for example a charged carboxylate group. Superstar is used to generate or to get a knowledge-based pharmacophore generation and prediction of intermolecular interactions. It uses the crystallographic information about the non-bonded interactions to generate the interaction map within the protein binding site or around the small molecule. Superstar retrieves its data from isostar. GOLD is a robust package for protein ligand docking using a genetic algorithm. GOLD stands for Genetic Optimization for Ligand Docking. All these modules or the tools are being maintained by CCDC. In addition to several databases which are under CCDC, CCDC also provides a CSD Java Sketcher, CSD Unity and CSD MDL and several other data updates. CSD provides a comprehensive and fully retrospective coverage of the published literature where one can have a full confidence that the CSD searches are returning all the crystal structure matches. Every crystal structure that has been deposited in CSD undergoes an extensive validation and cross-checking by expert chemists and crystallographers. CSD is enriched with various informations as we saw already that it contains a bibliographical information, the structural data, the chemical and the physical properties of the molecule. Information stored in the CSD for each entry can be considered in three different classes. First, there is a text-based information that contains bibliography, chemical name, formula, experimental information about the crystal structure determination, the procedure, and also the information about the shape, color, and the applications of the compound. Second, there is a chemical connectivity information in a form of 2D structural diagram, which is a basis of much of the sophisticated search missions for the CSD system. Third, there is a crystallographic information that consists of unit cell dimensions, space group, atomic coordinates, and so on. The following are the different information that is available in the CSD, like the compound name, chemical formula, chemical diagram, the property, the absolute configuration, the conformers, isomers, etc. Moving on to the text information, it contains details about the disorders, whether anything are present, the residual errors, the non-standard experimental conditions, and also about the polymorphism and so on. CSD is continuously updated with new structures. In addition to searching published crystal structures, one can also build their own local structure repositories that are then searchable either independently or in conjunction with CSD. CSD provides an ideal platform for making the students and researchers understand 3D structural chemistry. Visualization and interactive manipulation of real three-dimensional molecular structures helps in understanding the structural aspects. Data mining with CSD provides easy access to real measured data, complete with experimental errors and statistical variation that gives an opportunity to deal with certain uncertainties in structural chemistry and also helps in analysis of the structural data. CSD analysis are very important in structure correlation studies that includes a mapping of reaction pathways from crystal structure data and correlations of crystal structure geometry with ab initio calculations. The validated and curated CSD files can be exported into a wide range of formats. To name some formats are CIF, mole format, PDB, shell Excel or XMOL. One can visualize the structures by using any of the visualization programs like PyMol, Mercury, Chimera, Rasmol, etc. In addition to the analysis of the structural aspects for the data that are deposited in CSD, several statistical analyses can also be performed. To name a few, one can consider the examples of total number of organic structures, the structures that are solved by XRD, the number of structures for which the absolute configuration was determined, the number of structures with disorders, the number of structures which have the R factor less than 0.05, or the number of structures that belong to a triclinic system and so on. One can also generate a table showing the number of entries in the CSD, what are the different number of compounds that have been deposited, how many organic structures and organometallic structures are deposited, and how many structures are deposited with R factor less than 0.05 like we saw in the previous slide. One can also analyze the statistics like how many structures have been deposited in a particular system or in a particular space group. CSD R factor statistics can also be done. The precision of crystal structure determination is often assessed using the R factor. R factor is a measure
of how well the structure factors computed using the refined structural model agree with the structure factors given by the experimentally observed diffraction intensities. Lower the R value, better is the model.
Inorganic crystal structure database is used for deposition of inorganic compounds. ICSD is a comprehensive collection of crystal structure data for inorganic compounds such as salts, silicates, oxides. Web-based searching by empirical formula, compound name, and also by the crystal data, one can retrieve the data of the structural details of inorganic compounds. International Center for Diffraction Data is a non-profit scientific organization that promotes the applications of materials characterization methods in science and technology by providing different forums for the exchange of ideas and the information.
Powder diffraction file is a collection of single-phase X-ray powder diffraction patterns in a form of tables of characteristic interplanar spacings and corresponding relative intensities along with the physical and crystallographic properties of the particular molecule. PDF includes data originating from ICDD sources and also with collaborations with ICDD, FIZ and NIST. The compilation includes metals, minerals, organic and inorganic compounds.
Moving on to the deposition of macromolecular structures, Protein Data Bank is an archive of experimentally determined three-dimensional structures of biological macromolecules. It began in the year 1970s at Brookhaven National Laboratory under the leadership of Walter Hamilton, and now it is the world's largest repository for the processing and distribution of three-dimensional structural data of proteins and also the nucleic acids. In the year 1998, Research Collaboratory for Structural Bioinformatics, which is generally called as RCSB, took the responsibility for the management of Protein Data Bank. In the year 2003, Worldwide Protein Data Bank was formed to maintain a single PDB archive of macromolecular structural data that is freely and publicly available to the global community. It consists of organizations that act as deposition, data processing, and distribution centers for the protein data.
This shows the various sites that are maintained by WWPDB. The sites are BMRB, PDBE, PDBJ, and RCSB PDB. BMRB collects NMR data from any experiment and captures assigned chemical shifts, coupling constant, and peak list for a variety of macromolecules, and it contains derived annotations such as hydrogen exchange rates, pK values, and relaxation parameters. Protein Data Bank Europe. It contains rich information about all the PDB entries. Multiple search and browsing facilities are also available. The advanced services include PDBE PISA, PDBE Fold, PDBE Motif, advanced visualization and validation of NMR and EM structures. There are also certain tools available for bioinformatics work. PDBJ represents Protein Data Bank Japan. It supports browsing in multiple languages such as in Japanese, Chinese, and Korean language. RCSB stands for Research Collaboratory for Structural Bioinformatics. It is dedicated to improve the understanding of the function of biological systems through the study of three-dimensional structure of biological macromolecules. Understanding the shape of a molecule, a three-dimensional structure of a molecule, will give a clear picture about what is the function of the molecule. So there is always a structure-function relationship. Structure-function relationship analysis play a major role in drug development. Structures in the archive range from tiny proteins and bits of DNA to complex molecular missions like the ribosomes. From the RCSB PDB website, the structure data can be downloaded and the sequence can be downloaded in a FASTA format and also in a text format. One can also download the biological assemblies if any. RCSB PDB also explores the structure and function relationship of a new protein every month, and it is called as a molecule of the month. PDB also has monthly newsletters that includes the updates from the PDB as well as the articles about how people are utilizing the PDB for various projects. There are several references to the scientific events like the Science Olympiad protein modeling event will also be listed. PDB also has certain educational resources. It offers various tools to explore the molecules of biological processes. One can go through the background information on using the PDB, as well as there are certain lesson plans and some tutorials are also available. There are also tutorials that cover various topics like enzymes, protein synthesis, health and disease, what is the infrastructure and communication of biology and nanotechnology, and about the biological energy and so on.
RCSB Protein Data Bank has several regular updating features, like they update every month what is the molecule of the month, they repeatedly conduct the conferences and competitions. They also generate a tabular reports, the specialized visualization tools, the sequence and structure comparisons, as well as some interesting newsletters, and they have a dedicated educational resource which can be obtained from the RCSB database.
PDB structure ID number. It is also called as a PDB ID or PDB code. It's a 4-character alphanumeric identifier that is assigned to a particular data that is deposited to this database. It contains the information about the name of the molecule. It contains information about the authors or the researchers who were involved in purifying, crystallizing, and solving the structure. One can also check using what method the structure has been determined, either by using X-ray crystallography or by NMR or by electron microscopy. Resolution is a term that indicates how accurately the position of each and every atom was determined. Lower the number of resolution, the better is the data. That is, the smaller the number, better is the data, higher will be the resolution. Molecular description is another term one has to know. It indicates the number of chains within the molecule and also the chain identity. For example, in a case of a hemoglobin, there are 4 chains. Hemoglobin is a tetramer. A and C are alpha globin molecules and chains B and D are beta globin molecules. These kind of classifications will be given in detail in PDB. One can also check the functional class of the molecule and what is the source of the molecule, from which species the molecule has been isolated, whether it is from human, bacterium or virus. In order to overcome the objections, if any, being raised at the time of publication or at the time of depositing the data to the PDB, authors should always maintain the original frames of data that have been collected for macromolecule. This chart clearly shows how the PDB has evolved, the history of PDB, and also some structures or novel structures that are deposited in the PDB, and some of the structures have won the Nobel Prizes. Protein Data Bank was started in the year 1971. In 1973, there are the early structures that are deposited in PDB are carboxypeptidase, chymotrypsin, cytochrome B5, hemoglobin and myoglobin, rubredoxin, subtilissin, trypsin inhibitor, lactate dehydrogenase. In the year 1989, IUCN made a policy on data depositions that is to be published. In 1998, RCSB has took the in charge of PDB, and in 1999 the 10,000th structure has been deposited and released. In 2002, EMDB established at MSD EBI. In 2003, worldwide PDB was established by RCSB PDB, PDB-E and PDB-G. In the year 2006, BMRB has joined with worldwide PDB. In 2008, the 50,000th structure has been deposited and released. From 2010 onwards, deposition of chemical data shift has been made mandatory in a PDB. In 2011, PDB has celebrated 40th anniversary and they conducted a symposium called PDB 40 Symposium. At that time, more than 77,000 structures have been deposited and released from the PDB. To move on further with some of the interesting and important structures that have been deposited in PDB, the first tRNA structure that deposited in PDB was in 1973. In 1979, the first Z-type DNA was determined. In 1981, the first bDNA structure was determined by Drew et al. In 1982, Clark received the Nobel Prize for the development of crystallographic electron microscopy and also for the discovery of biologically important nucleic acid and protein complexes. In 1988, Desenhofer, Huber, and Michel determined the three-dimensional structure of a photosynthetic reaction center, and the first NMR entry to be deposited in PDB was in the year 1989. The first EM entry was made in 1991. In the year 1997, Boyer, Walker, and Scoop received the Nobel Prize for their studies on enzymatic mechanism that underlie the synthesis of adenosine triphosphate and the discovery of iron transporting enzymes. The first ribosome structure was deposited in the year 2000. In 2002, Utrecht received the Nobel Prize for the development of NMR spectroscopy for determining the 3D structures of biological molecules in solution. In 2003, McKinnon deposited the structure regarding to the potassium channels, and he received the Nobel Prize in 2003. In 2006, Kornberg received the Nobel Prize for the studies on molecular basis of eukaryotic transcription. Nobel Prize in 2009 has been awarded to Ramakrishnan, Straits and Leonard for their work on the structural studies of ribosome and also for the functional studies of the ribosome, and they have deposited large number of ribosome structures in the PDB. One can prepare a chart on the statistics of the PDB current holdings, like where you can categorize how many protein structures are deposited, how many nucleic acid structures are deposited, or protein nucleic acid complexes or protein ligand complexes has been deposited, and what experimental method has been used to solve the structure, either by X-ray crystallography or NMR or electron microscopy and so on. Various segments of classifications are used in RCSB PDB, and some are shown here. Like the organism can be characterized, the taxonomy, what experimental method, if the data has been collected by using X-ray diffraction, then what is the resolution of the data, and the enzyme classification, the protein symmetries, if any they are present, and how many membrane proteins are deposited in the PDB, membrane protein structures are deposited in the PDB. All these classifications one can go through. This clearly shows the yearly growth of total number of structures that are being deposited in a PDB, the number of protein, DNA, RNA structures, or the number of protein nucleic acid complexes are deposited. One can prepare a chart for that. It is very clear that there is an exponential increase in the number of structures that are being deposited in the PDB. Due to the advent of synchrotron radiation, the number of structures deposited in the PDB is increasing enormously since 1980s. As on today, average of about 180 structures are being deposited every week. Nucleic acid database is again an important database. It is found in the year 1991. It is used to assemble and distribute the structural information about the nucleic acid. In addition to the primary structural data that contains in PDB, NDB contains the annotations that are specific to nucleic acid structure and function, as well as tools that enable the users to search, download, analyze, and also learn more about the nucleic acids. NDB is a value-added database that provides the services specifically for the nucleic acid community. As on 23rd March 2016, 8,062 structures have been released in nucleic acid database. One can do different types of searches, like one can search the DNA, search the RNA, and also one can do an advanced search based on the chemical features, the binding mode, and based on the experimental information. Nucleic acid database also has certain kind of featured tools like RNA 3D motif atlas, which is a representative collection of RNA 3D internal and hairpin loop motifs. R3D align is an application for detailed nucleotide to nucleotide alignments of RNA three-dimensional structures. ChemNet Base is a collection of chemistry database from CRC Press, which can be subscribed to individual or collectively. It maintains again a set of dictionaries or libraries, like for example Handbook of Chemistry and Physics, Combined Chemical Dictionary, Dictionary of Natural Products, Organic Compounds, and so on. Let's move on to Combined Chemical Dictionary. This dictionary is a structured database that contains detailed information on chemical substances. It includes descriptive and numerical data on chemical, physical, and biological properties of the compounds. Also, the systematic and common names of compounds are given. The literature references, the diagrams, and their associated connection tables will also be given. It includes all currently marketed drugs, including all those that are listed in generic name compilations, the US adopted names, international non-proprietary names, British approved names, Japanese accepted names, and so on, as well as those undergoing clinical trials, the compounds with an established use such as the catalyst, solvents, synthetic reagents, analytical reagents, everything will be deposited here. With a vast range of data for both small and macromolecules that are available, one can do the search in different ways.
Every data that is deposited in protein data bank contains information about the atomic coordinates, primary and secondary structure information, sequence database references, bibliographical information, biological assembly, and also details about the data collection. What are the methods that are used to solve the structure and define the structure? Each PDB file will receive a 4-letter code, which is generally an alphanumeric identifier. Every PDB file in a protein data bank has its own page which contains a lot of information like what we saw in the previous slide. So students, let's now summarize what we have learned in this module on crystallographic databases. There are several databases we saw that are available for the deposition of structural details of both small and macromolecules, and also in small molecules for organic, inorganic, and organometallic compounds, and also the metal compounds. Cambridge Structural Database and Protein Data Bank we saw in more detail. Cambridge Structural Database is a world repository for small molecule organic and organometallic crystal structures, whereas the PDB is used for the deposition of macromolecular crystal structures. CSD is widely used resource in structural chemistry, which is maintained by Cambridge Crystallographic Data Center. It provides a lot of other services which we saw elaborately in the module. And regarding the protein data bank, it is maintained by RCSB. It provides information and more about the macromolecular structures of proteins, as well as the protein ligand complexes and also protein nucleic acid complexes. It is the largest worldwide repository. Thank you.